protection is never passive. It is an active, adaptive negotiation with threat. Welcome back to the Deep Dive.
Hello again.
Today we are stepping into a world that is, frankly, mind-bogglingly complex. It might be the most chaotic, intricate, and honestly beautiful system in the body outside of the brain itself.
It really is.
We're talking about the immune system.
Chaos is actually a very good word for it. I mean,
Yeah.
if you were to just look at a slide of inflamed tissue without understanding the rules, it looks like a riot. It looks like complete anarchy.
Right.
But once you understand the physiology, the underlying logic, it's a symphony.
A symphony with assassins.
A symphony with assassins, yeah.
Yeah.
And landmines and incredibly strict rules of engagement.
I like that. So for today's deep dive, we're focusing on Physio 03, immunity, infection, and inflammation.
A small topic.
Just a light one.
Yeah.
And to guide us, we're pulling from the two giants, the two Bibles of medical physiology. We've got Ganong's review of medical physiology, and we're also weaving in the perspective from Guyton and Hall.
And it's really important to understand why we use both. They're not redundant. Ganong gives us that beautiful mechanistic depth.
The nitty-gritty.
The nitty-gritty, the molecules, the receptors, the signaling pathways inside the cell.
Yeah.
But Guyton gives us the so what,
The big picture.
the systems level coherence. How does this symphony connect to blood pressure, to kidney function, to homeostasis for the whole organism?
And that brings us to our mission for today. We have a very specific goal.
Yes.
We are not going to just list cells. I think everyone listening has had that biology class. Here's a neutrophil, here's a macrophage, here's a T cell. Memorize their functions for the exam.
Right, the laundry list approach.
We want to get past that. We want to understand the strategy, the logic.
The why. Because if you understand the logic, you don't actually have to memorize nearly as much. It makes sense.
Which brings us to the central metaphor we're going to be using today, Medlock Holmes.
Medlock Holmes. I love it because it reframes the entire problem. We have this tendency, to think of the body as a fortress. Exactly. A castle with stone walls, a moat, the drawbridge is up. The goal is to keep everything out.
But that's not how it works.
It can't work that way. Physiology teaches us that the body is fundamentally an open system.
What do you mean by open?
Well, you have to eat, you have to breathe, you have to interact with the world, your gut, your lungs, your skin. These are vast surface areas, hundreds of square meters, that are in constant direct contact with the outside world.
So it's not a fortress at all.
It's more like a bustling port city, a city with open borders where millions of entities are coming and going every single second.
And if the borders are open, that means some shady characters are always walking in.
Always. Bacteria, viruses, fungi, parasites, toxins, they are walking in with every breath you take, with every meal you eat. So the core problem for the immune system isn't building a higher wall.
It's identification.
It is detective work, it's intelligence.
It's Medlock Holmes walking the beat.
Exactly. The detective has to look at every single face in the crowd, every protein, every molecule, every cell, and ask the hardest question in all of biology.
Are you one of us? Or are you one of them?
Is this self or is this non-self? And it gets even harder than that.
How so?
Because even if it's non-self, that doesn't automatically mean it's a threat.
Right. Is it a harmless tourist or is it a terrorist with a bomb?
Precisely. That piece of peanut protein you just ate, that's non-self. That pollen grain you just inhaled, non-self. The trillions of helpful bacteria in your gut, all non-self.
So if Medlock Holmes gets it wrong,
the consequences are catastrophic. If he shoots the tourist, the peanut protein, you get an allergic reaction, or worse, anaphylactic shock.
and if he shoots one of his own, a self cell,
If he mistakes your own pancreas cells for an enemy, you get type 1 diabetes. If he attacks your joints, you get rheumatoid arthritis. That's autoimmune disease.
well, if he fails to shoot the actual terrorist,
You die of overwhelming infection.
the stakes couldn't be higher.
They are absolute, and that is the tension we are exploring today. How does the body make that life or death decision millions upon millions of times a day without destroying itself in the process?
Okay, so let's start by laying out the precincts. The police force is generally divided into two main branches.
Correct. You have the innate immunity and the adaptive, or sometimes called acquired immunity.
And I really want to spend some quality time on the innate system first. In our outline, you called them the ancient watchmen. Why ancient?
Because this system is evolutionarily ancient. It is the bedrock of multicellular survival on this planet. We share these fundamental defense mechanisms not just with other mammals, but with fruit flies, with starfish, Even plants have a recognizable innate immune system.
Plants, really?
Oh, absolutely. A tree doesn't have antibodies or T cells, but it absolutely knows when a fungus is trying to digest its roots, and it can mount a localized chemical defense. This system is that old.
So what defines it? What's its core characteristic?
The key concept is that it's germline encoded.
Okay, unpack that phrase for us. It sounds like a textbook term, but it's important.
It is. Germline encoded means the instruction manual for these defenses is written directly into the DNA that you inherited from your parents. It's hardwired, it is fixed from the moment of conception, and it does not change throughout your life.
So my innate immune system today is functionally identical to the one I had the day I was born.
Yes, it doesn't learn from experience. It doesn't go to school. A neutrophil, which is a key foot soldier of the innate system, is born knowing how to recognize and eat a bacterium. It doesn't need to be trained. It is born ready for action.
Okay, but that raises a huge question.
Yeah.
If it doesn't learn, and there are millions of different species of bacteria and viruses out there, how on earth does it know what a bad guy looks like?
This is the absolute genius of the innate system. It's incredibly clever. It doesn't waste time trying to recognize specific individuals.
It looks for uniforms.
It looks for uniforms, exactly. The technical term is pattern recognition. The cells of the innate system are covered in what we call pattern recognition receptors, or PRRs.
And they're scanning for what kind of patterns?
They scan for molecular structures that are, 1, essential for the life of broad classes of microbes, and 2, are never found in human tissues. We call these patterns PAM-MPs, pathogen-associated molecular patterns.
Okay, give me a concrete example. What's a classic uniform that a bacterium wears that a human cell never would?
Peptidoglycan. It's a key component of the bacterial cell wall. It's this rigid, cross-linked mesh of sugars and amino acids that gives bacteria their shape and stops them from bursting.
In our cells?
Our cells have soft, flexible, lipid-based membranes. We simply do not make peptidoglycan. There is no gene for it in our entire genome. So if an immune cell detects peptidoglycan and knows with absolute 100% certainty this is not us, there's no ambiguity.
That's like a molecular barcode scanner.
That's a perfect analogy. And these scanners, these receptors, have a fantastic discovery story behind them. This brings us to the Toll story.
Yes, I love this one. It's one of the few genuinely funny names in molecular biology.
It is. So back in the 1980s, Christian Nusslein-Volhard and Eric Wieskhaus were doing this landmark work on fruit fly development, trying to figure out which genes control the body plan of the larva.
And they were just mutating genes at random to see what happened.
Exactly. And they found one mutation that caused the larvae to develop all messed up. Their ventral, or underside, was underdeveloped.
So the German researcher looks at this weird little larva under the microscope and says,
"Das ist ja toll." Which in German can mean, that's amazing, but in that context, it was more like, wow, that's weird, or that's crazy. So they literally named the gene Toll.
But at the time, they thought it was purely a developmental gene. It had nothing to do with immunity.
Correct. The big leap came years later when a different group, led by Jules Hoffman, discovered that adult flies with a mutated Toll gene were incredibly susceptible to fungal infections. They were dying left and right.
So the same gene that patterned the larva was also fighting fungus.
That was the connection. The Toll gene didn't just build the fly. It coded for a receptor that was crucial for recognizing and fighting off fungal pathogens. That discovery opened the floodgates.
And then we found them in humans.
We found a whole family of them, and because we're not as creative, we just called them Toll-like receptors, or TLRs.
Okay, so this is where we need to get into the weeds of Genong a bit, because as you said, understanding which TLR sees which barcode explains a huge amount of clinical medicine.
It really does.
So let's start with the celebrity, the one that's always in the headlines. TLR4.
TLR4 is the rock star of the family. Its claim to fame is that it recognizes lipopolysaccharide, LPS.
And just to refresh everyone's memory from microbiology, what is LPS?
LPS, also known as endotoxin, is a major component of the outer membrane of gram-negative bacteria. Think E. coli, Salmonella, Pseudomonas, Neisseria. It's part of their skin. It's not a weapon they actively shoot at us. It's just part of their structure.
So why is TLR4's ability to see this so critically important?
Because of the sheer magnitude of the response it triggers. When TLR4 on the surface of a macrophage or a dendritic cell binds to LPS, it doesn't just send a polite note to the nucleus, it pulls the fire alarm for the entire building.
And this is the mechanism behind septic shock.
This is the central mechanism of septic shock from gram-negative bacteria.
Let's slow down and walk through that because that's a huge clinical point. Sepsis is when the body's shutting down, right?
Yeah.
Blood pressure plummets, organs start to fail.
Exactly. And for a long time, the thinking was, oh, the bacteria are just overwhelming the body, eating the organs. But that's not really what's happening. Septic shock is a disease of our response. It's friendly fire on a massive scale.
It's the immune system overreacting.
It's the TLR4 system going into absolute panic mode. It sees LPS in the bloodstream, concludes the body is being invaded on a massive scale, and triggers the release of an overwhelming flood of inflammatory cytokines.
Things like TNF-alpha and interleukin-1.
They big guns. And these cytokines cause systemic vasodilation. All the blood vessels in your body relax and get leaky at the same time.
So the container gets bigger, but the amount of fluid stays the same. The pressure has to drop.
The pressure crashes. Your organs are no longer perfused with blood. And you die. You die from your own defense mechanism, not directly from the invader. That is the power and the danger of the TLR4 pathway.
That is terrifying. It's that theme you always bring up, regulation versus capacity. We have the capacity to kill ourselves in our own defense.
And we do it all the time. Sepsis is a leading cause of death in hospitals. And it's all because this ancient life-saving system lacks a proportional response switch when the threat is systemic.
Wow. Okay, let's look at a few other key TLRs. What about TLR5?
TLR5 is what I like to call the tail hunter.
The tail hunter.
It specifically recognizes a protein called flagellin.
Which is what makes up the flagellum, the little propeller tail on motile bacteria.
Right. And again, it's a perfect pattern. Human cells don't have flagella. We don't swim around with protein tails. So if TLR5 sees flagellin, it knows this is an invader.
Simple and effective. What about TLR9? You mentioned that one is a bit different.
It is, because TLR9 isn't on the cell surface. It's an intracellular scanner. It lives inside a compartment called the endosome.
So it's looking for things that have already been brought inside the cell.
Exactly. It's looking for enemies that have already breached the first wall. And its target is bacterial DNA.
But hold on, we have DNA. How does it tell the difference between our DNA and bacterial DNA?
That's the cool part. It comes down to a subtle chemical difference. Bacterial DNA has a high frequency of these specific sequences called CpG motifs, where the cytosine base is unmethylated.
And in our DNA?
In mammalian DNA, these CpG motifs are much rarer, and they're typically methylated. They have a little chemical cap on them. TLR9 is exquisitely tuned to recognize the unmethylated form. It can literally taste the difference in the genetic code itself.
So it's like a spell checker looking for a specific typo that only appears in the enemy's documents.
What a great way to put it, yes. And there are others. TLR2 recognizes lipoproteins from gram-positives. TLR3 sees double-stranded RNA, which is a classic sign of a virus. The point is, the innate system has this whole toolkit of scanners for different microbial uniforms.
Okay, so the scanner beeps. Medlock Holmes has seen the peptidoglycan or the LPS or the flagellin. He's identified the suspect. What happens next inside the cell?
Now we get the signal transduction. This is the part Genong explains so well. The receptor on the outside has to communicate the threat to the command center on the inside, which is the nucleus.
It has to send a runner with a message.
The most famous runner in this story is a transcription factor called NF-kappa B.
NF-kB. It sounds like a secret agency, but I know it's a huge deal in inflammation.
It is arguably the master switch of inflammation. In a resting happy cell, NF-kappa B is essentially held hostage in the cytoplasm. It's bound up by an inhibitor protein called II, so it can't do anything.
It's locked in a closet.
Exactly. But when TLR4 binds to LPS, it sets off a chain reaction of kinases enzymes that stick phosphate groups onto things, and this leads to the destruction of the inhibitor.
The lock on the closet is broken.
The lock is broken, NF-kappa B is free. It immediately translocates, rushes into the nucleus, binds to the DNA at specific promoter regions, and acts like a power switch, turning on hundreds of genes for war.
And what does war look like at the chemical level?
At the chemical level, it looks like cytokines.
We hear that word all the time. Cytokine storm became a household phrase recently.
Yeah.
But in plain English, what is a cytokine?
A cytokine is a radio broadcast. It's a small, soluble protein that is released by one cell to change the behavior of other cells.
It's a command.
It is a command. Interleukins, like IL-1 and IL-6, interferons, tumor necrosis factor, or TNF, these are all cytokines, and they are carrying orders. Come here. That's a chemokine. Eat this. That's an opsonin. Make the blood vessel leaky so reinforcements can get in. Turn up the body's thermostat and cause a fever.
So the innate system sees the pattern, frees the NF-kappa B runner, who tells the nucleus to print out millions of radio orders or cytokines.
Exactly. And the result of all those orders is what we recognize clinically as inflammation. Redness, swelling, heat, and pain. And, crucially, the arrival of the professional eaters.
The phagocytes.
The phagocytes, the neutrophils, which are the first responders, the SWAT team, and the macrophages, which are the bigger, slower-moving cleanup crew. They follow the chemical trail of cytokines right to the site of infection and just start physically engulfing and destroying the enemy.
It all sounds incredibly fast and effective.
It is. It is stunningly effective. It handles probably 99% of the microbial threats you encounter every single day without you ever knowing it. That little paper cut that got a bit red yesterday and is fine today, that was your innate immune system winning a war you never even noticed.
But there's always but.
There's a big but. For all its speed and power, the innate system has a fatal flaw.
It's dumb.
Dumb is a little harsh. Let's say it's generic or indiscriminate.
It uses a shotgun.
It uses a shotgun, exactly. It treats all gram-negative bacteria the same way because all it sees is the LPS uniform. But some enemies are much more devious than others. Some have evolved ways to hide from the scanners. Some hide inside our own cells where the phagocytes can't reach them. The shotgun approach isn't always enough.
And that's when you need the sniper.
That's when you need the sniper. Enter Section 2, Adaptive Immunity.
The strategist and the archivist. The text mentions a surprising fact about where this system came from. It wasn't a slow, gradual evolution.
No, this is one of the most remarkable, almost unbelievable events in evolutionary history. All the evidence suggests that the entire machinery for adaptive immunity appeared very suddenly, about 450 million years ago, right around the time of the first jawed vertebrates, like early sharks.
And it was an accident.
It looks like a happy accident. The leading theory is that a virus infected one of our distant ancestors and inserted a piece of its own genetic material into our ancestor's genome. Specifically, it inserted a type of jumping gene called a transposon.
A transposon.
It's a piece of DNA that can cut itself out of one part of the genome and paste itself into another.
Usually that sounds like a very bad thing. Randomly cutting and pasting your DNA sounds like a recipe for cancer.
It is usually very bad. But in this one specific instance, the genes that got inserted, which we now call the recombination activating genes, RUG1 and RG2, gave a specific lineage of cells a superpower.
The power to shuffle their own DNA.
To intentionally and precisely shuffle specific segments of the genes that code for their receptors.
Let's try to put that in perspective.
Yeah.
The innate systems instruction book is written in pen. It's fixed.
It's fixed. You can only read what was printed at the factory.
The adaptive system.
The adaptive system was given a bag of Scrabble tiles, and the RAG enzymes are the player who can reach into the bag and arrange those tiles to spell literally any word imaginable.
Even words that have never existed before.
Especially words that have never existed before. This process, which is called VDJ recombination, allows your body to generate a vast, almost infinite repertoire of B-cell and T-cell receptors. It can create receptors that recognize synthetic chemicals made in a lab. It can create receptors that could recognize a virus from Mars.
It's a system designed to recognize the unknown.
That is the perfect way to describe it. It's future-proof. And this incredible diversity is the foundation for the two main arms of the adaptive system.
Humoral and cellular.
Right. Humoral is an old term for things found in the humors or body fluids. This refers to B lymphocytes or B cells.
And their weapon is the antibody.
The antibody, which is basically a secreted, soluble version of their receptor. They mass produce these Y-shaped proteins and release them into the blood and lymph.
They're like heat-seeking missiles.
So maybe more like smart handcuffs. They float around, find their one specific target, a toxin, a virus particle, and latch onto it, either neutralizing it directly or tagging it for destruction by the phagocytes.
And the other arm, the cellular arm.
That's the domain of the T lymphocytes, or T cells. Their receptors stay bound to their own cell surface. They don't shoot missiles.
They engage in hand-to-hand combat.
Some of them do, yes. Those are the killer T cells. Others are the generals, the orchestrators. They physically touch other cells to give them orders.
Okay, so this brings me to a math problem I've always struggled with. You said we can generate this almost infinite library of receptors.
Yes.
But the body is finite. I only have so much space. So for any given specific target, like one particular strain of the flu virus, I must only have a tiny number of cells that can actually recognize it, right?
That's absolutely right. At any given moment, you might have only a handful, maybe one or two of naive T cells in your entire body that have the perfect receptor for that specific flu virus.
So if I get infected and there are billions of virus particles replicating in my lungs,
Yeah.
how does that one single cell fight them off? It's impossible.
It is impossible. It can't, not on its own. And this is where we get to the absolute core principle of the adaptive response. It's called clonal expansion.
Okay, walk me through this. The virus gets in.
The virus gets in.
Yeah.
Your innate system immediately goes to work. The watchmen are fighting. They're dying. They're releasing cytokines. They are holding the line and, crucially, buying time.
And while that's happening,
The alarm has gone out, and the body is frantically searching through its entire library of naive B cells and T cells, looking for that 1 in a billion match.
it's like checking every single ID badge in a city of millions.
It is. A dendritic cell, which we'll talk more about later, carries a piece of the virus to the local lymph node and shows it to T cell after T cell. Is this yours? Nope. Is this yours? Nope. Is this yours? Aha.
And when it finds the match,
When that one specific lymphocyte finds its cognate antigen, the one thing in the universe it was born to recognize, it's like a switch flips. It receives the signal to activate. It shifts from a quiet, resting patrol mode to an explosive production mode.
it starts to clone itself.
Proliferates wildly. 1 cell becomes 2. 2 become 4. 4 become 8. It starts dividing every few hours. Within about a week, that single solitary cell has produced an entire array of millions of identical clones, all armed with the exact same receptor, all targeted at that one specific flu virus.
And that delay, that's why we feel sick.
That explains the lag time. Why does the flu knock you out for 5 to 7 days before you start to feel better? That's the time it takes to select and build the clonal army.
So the fever, the aches, the fatigue, that's the innate system's desperate hold in action. And the recovery when you finally turn the corner.
That's the arrival of the specific adaptive clonal army. They sweep through the body and eradicate the threat with ruthless efficiency.
It's a beautiful logical system. But what happens after the war is won. The army can't just stick around forever.
No, that would be dangerous and energetically wasteful. The vast majority of the newly made effector cells, the soldiers, are programmed to die. They undergo apoptosis or programmed cell death to clear the battlefield.
But not all of them.
Not all of them. A small platoon is left behind. These are the memory cells,
The archivists.
the archivists, memory B cells and memory T cells. And these cells are different. They're long-lived. They can persist for decades, maybe even for your entire life. And they're easier to activate than the naive cells were.
So if that same flu virus tries to invade again 10 years later,
You're not starting from 1 cell. You're starting from a pre-existing battalion of maybe 10,000 veteran cells. The search phase is skipped. The cloning is faster and more robust. The response is so prompt and magnified that you crush the invasion before you even develop symptoms.
you're immune.
You're immune. And that right there is the entire physiological basis of vaccination.
We're basically creating the memory platoon by showing the body a wanted poster instead of having it fight the actual criminal.
That's a perfect analogy. We trigger the primary response safely, so the memory is in place for when the real threat arrives.
Okay, so we've met the players. Innate, the shotgun. Adaptive, the sniper. But where do these cells actually come from? Let's get into section 3, the developmental journey.
The origin story.
And like so many things in hematology, it starts in the bone marrow.
Everything starts in the bone marrow. With the hematopoietic stem cell, the mother of all blood cells, it churns out the common lymphoid precursors. But then, for these cells, there comes a critical fork in the road.
A destiny-defining moment for a young lymphocyte.
It really is. If that precursor cell stays in the bone marrow to mature, it will become a B cell.
B for bone marrow. That's a helpful mnemonic.
It is. Historically, the B actually stood for the bursa of Fabricius, which is an organ in birds where B cells mature, but for humans, bone marrow works perfectly.
And what if it doesn't stay home? What if it leaves the bone marrow?
If it leaves the marrow and migrates through the bloodstream to a small gland sitting just behind your breastbone called the thymus, it will become a T cell.
T for thymus. Simple enough. Now, I think most people picture the thymus, if they picture it all, as this sort of quiet, fatty little gland that shrinks away after puberty. But you describe it as a boot camp.
It is the most rigorous, most brutal boot camp on the planet. I would call it a slaughterhouse.
A slaughterhouse. Why? What's so dangerous about the thymus?
To understand that, you have to remember the RAG genes and VDJ recombination. We said this process creates random receptors.
Right, and almost infinite diversity.
Well, random is a double-edged sword. If you randomly generate a T-cell receptor that recognizes a protein from the measles virus, that's fantastic. That's a useful soldier. But what if, by pure chance, you randomly generate a receptor that binds with high affinity to your own insulin?
Ooh.
Or to a protein in the myelin sheath that insulates your nerves?
Then you've just created an assassin that is perfectly programmed to destroy a vital part of its own host. You've created a seed for autoimmune disease.
Exactly. So the primary and absolutely critical job of the thymus is to weed out these self-reactive T cells before they are ever released into the body. This process is called thymic selection, or T cell education.
And it's a two-step exam.
It is. The young T cells, called thymocytes, first undergo positive selection. This happens in the cortex, the outer part of the thymus. There, specialized epithelial cells display our own self-proteins on a molecule called MHC, which we'll talk more about.
What's the test?
The test is, can you even see this? Can you bind weakly to our own MHC? If a T cell's receptor is so malformed that it can't recognize self-MHC at all, it's useless. It can't receive signals, so it's instructed to die. It fails by neglect.
You're incompetent. You can't even see the ID badge. Get out.
Exactly. But the survivors of that test then move deeper into the medulla of the thymus for the big one, negative selection.
This sounds more ominous.
It is. Here, another amazing set of cells, the medullary thymic epithelial cells, have a superpower enabled by a gene called AIR. They can produce and display thousands of proteins from all over the body. Proteins from the eye, from the liver, from the pancreas, from the skin.
So they create this sort of molecular hall of mirrors of the entire body.
It's a pop quiz on the entire self-proteome. And they show these self-antigens to the surviving T cells. And the question is simple. Do you bind to any of these?
And if the T cell binds strongly, if it says, ooh, that pancreas protein looks tasty, I'd like to attack that.
It is immediately given a signal to commit suicide, apoptosis. It is executed on the spot.
That's brutal.
It has to be. And the numbers are staggering. Over 98% of the T cells that enter the thymus never make it out alive. They die there.
98%. That's an astonishing failure rate.
It's the price you pay for generating random diversity. You have to be incredibly stringent in filtering out the dangerous ones. Only that elite 2%, the ones that are competent but not self-reactive, are allowed to graduate and enter the circulation as mature, naive T cells.
So these graduates, these elite soldiers, finally enter the world. And the textbook makes a really interesting point that only about 2% of the body's total lymphocytes are actually in the bloodstream at any one time.
Right. We call them white blood cells, and we measure them in a blood test, but the blood is really just the highway. It's their commute.
So where do they actually live? Where are they stationed?
They live on base, in the secondary lymphoid organs, the lymph nodes, the spleen, the tonsils, the adenoids, the pairs, patches in the gut. That's where they congregate.
So the immune system is really a network of fortresses or surveillance outposts connected by these highways.
That's a great way to think about it. The blood and the lymphatic vessels are the highways. The cells are constantly circulating. They'll ride the bloodstream, then exit into a lymph node, percolate through it for a few hours, scanning for any intel.
Checking the bulletin boards.
Checking the bulletin boards. And if there's nothing going on, they'll leave via the efferent lymphatic vessel, which eventually dumps back into the bloodstream, and they'll ride the highway to the next lymph node down the line. They are constantly patrolling the entire network.
Which brings us perfectly to section 4, integration. Because so far we've kind of talked about these two systems, innate and adaptive, as separate entities, but they have to be in constant communication.
They absolutely have to be. They are deeply, deeply integrated. And this conversation solves what you could call the blind general problem.
What's the blind general problem?
Well, the T cells are the generals, the strategists, but they're mostly sitting back in the command center, the lymph node. If you get a splinter in your big toe, that T cell general sitting in the lymph node up in your groin has no idea it happened. It's blind to the local events out in the tissues.
It's too far away from the crime scene. So how does it get the news? Who tells the general that a war has started?
The scout, the informant, the cell that bridges the two worlds. The technical term is the antigen-presenting cell, or APC.
And the most important, the most professional APC is the dendritic cell.
The dendritic cell is the MVP. That's my favorite cell in the immune system. They're these beautiful star-shaped cells with long, spindly arms, and they live out in the peripheral tissues, in your skin, in the lining of your gut and lungs. and they are constantly sampling their environment.
They're like the food tasters for the king.
They are, a process called macropinocytosis. They're constantly sipping the extracellular fluid, grabbing proteins, debris, whatever is around them.
Just to see what's out there.
Right. Now imagine one of these dendritic cells in your toe gets that splinter. It picks up some bacteria from the wood. It immediately recognizes the bacterial PMPs, the barcodes, via its toll-like receptors.
So the innate scanner goes off.
The innate scanner goes off, and that does two things. First, it tells the dendritic cell what you just ate is dangerous. Second, it gives the cell a command. Leave your coast and report to headquarters.
It leaves the crime scene.
It literally pulls in its long arms, changes its shape, and begins to crawl out of the tissue. It finds a tiny lymphatic capillary and rides the lymphatic fluid like a water slide all the way to the nearest draining lymph node.
And it's carrying evidence.
It is carrying the crucial evidence. It has digested the bacteria it ate and has loaded little fragments of the bacterial proteins onto a special display molecule. This is the MHC, the major histocompatibility complex.
Okay, this is a term that trips up every single medical student.
Yeah.
MHC. Let's break this down as simply as possible.
Let's use an analogy. Think of the MHC molecule as a hot dog bun.
A hot dog bun. Okay, I'm with you.
The antigen, the little piece of the bacteria, is the hot dog. The T cell receptor is very, very picky. It can only recognize the hot dog if it is nestled snugly inside the bun. It cannot see free-floating hot dogs.
That is a very specific hunger.
It's an incredibly important safety mechanism. It ensures that T cells only get activated by antigens that are being formally presented to them by another one of our own cells. So the dendritic cell arrives in the lymph node, which is packed with thousands of different T cells, and it runs around shoving this hot dog bun in the face of every T cell it meets.
Is this yours? Did you order this hot dog?
Is this the one you're looking for? Until, by chance, it finds the one naive T cell whose receptor is a perfect match.
And we should probably quickly distinguish between the two types of bun, right? Class 1 and Class 2 MHC.
Yes, that's a key point from Genome. MHC class I is the internal affairs bun. It's found on almost all the nucleated cells in your body. It's constantly displaying little bits of the proteins being made inside that cell. It's like a little window into the cell's factory floor.
So it's for showing what's happening on the inside.
Right. Here's what I'm making today. This is how we catch cells that are infected with a virus, because they'll start making viral proteins and displaying them in their class I MHC. This is the bun recognized by the CD8 killer T cells.
The assassins. In Class 2.
MHC class 2 is the external affairs bun. It's only found on the professional antigen-presenting cells, dendritic cells, macrophages, B cells. It's for displaying pieces of things they've eaten from the outside world.
Look what I found out there.
Look what I found. And this is the bun that's recognized by the CD4 helper T cells, the generals.
Okay, so the dendritic cell, our informant, has found the one matching helper T cell. The T cell receptor physically binds to the MHC antigen complex. Is that it? Does the war start now?
Not yet. And this is probably one of the most important safety checks in the entire system. It's called the two-signal hypothesis.
A fail-safe on top of the fail-safe.
Yeah. Absolutely. Signal 1 is the antigen match, the T cell receptor binding the MHC peptide complex. This is the enemy. But if a T cell receives signal 1 alone, without anything else, a strange thing happens. It doesn't activate. It becomes permanently shut down, a state we call anergy.
Wait, why would it do that? That seems completely counterintuitive.
Think about it. What if that dendritic cell wasn't in an infected toe? What if it was in a healthy gut and it just happened to pick up a harmless piece of a food protein or a piece of a friendly commensal bacterium?
Or even a piece of one of our own cells that just died of old age.
Exactly. You don't want to launch a full-scale military attack every time a cell presents an antigen. You need a second piece of information. You need confirmation that the antigen it's presenting is associated with actual danger.
So what is signal 2?
Signal 2 is co-stimulation. Remember when the dendritic cell first saw the bacteria with its toll-like receptor? That innate danger signal told it to do something else. It told it to put a second molecule on its surface, a molecule called B7. And the T cell has a receptor for that called CD28. This B7-CD28 interaction is signal 2. It's the confirmation. It's the dendritic cell saying, not only did I find this antigen, but I found it in a context of inflammation and microbial patterns. This is a real threat.
It's like the nuclear launch sequence. You need two different people to turn their keys at the same time.
That is the perfect analogy. Key 1, target acquired. Key 2, we have confirmed hostile intent. Only when both keys are turned simultaneously, antigen and co-stimulation, does the T cell truly activate.
And what does activation look like for a T cell?
It starts producing its own growth factor, a cytokine called interleukin-2, or IL-2. It puts out receptors for IL-2. This creates an autocrine loop it tells itself to divide, and that is the engine that drives clonal expansion.
That's when the army rises.
That is when the army rises. And then those newly activated helper T cells start producing other cytokines to orchestrate the entire response. They close the loop.
They start shouting orders back to the innate system.
Exactly. A Th1 type helper cell will release interferon gamma, which is a powerful signal that superactivates macrophages, telling them, become better killers, digest what you eat more effectively. A Th2 type helper cell might release IL-4 and IL-5 to tell B cells, we need antibodies, and we need to recruit eosinophils. They become the conductors of the entire symphony.
It's a stunningly complex and beautiful feedback system, but as we know, complexity is a breeding ground for failure.
It is.
Which brings us to our final section, the clinical case study. We're looking at Crohn's disease.
Crohn's disease is a perfect and tragic example of the system going wrong. It's a chronic, relapsing, and often destructive inflammatory disease of the digestive tract.
And the symptoms can be devastating. Ulcers, bleeding, severe abdominal pain, malnutrition, weight loss.
It's a terrible disease. And when we see that kind of intense inflammation, our first thought is always infection. There must be some horrible pathogen, some bad germ that's causing this. But the strange thing about Crohn's is that in many cases, we can't find a specific pathogen. The germs that seem to be driving the inflammation are just the normal commensal microbiota, the trillions of bacteria that are supposed to be living peacefully in our gut.
So Medlock Holmes isn't just arresting the wrong guy, he's arresting the local baker, the postman, the librarian.
Worse, he's burning down the whole town to get at them. This is, at its heart, a failure of tolerance. The immune system has lost its ability to peacefully coexist with the normal gut flora.
So what breaks? What goes wrong in the system to cause this?
The textbooks highlight a key genetic link, a gene called NOD2.
Wait, NOD2, we mentioned that briefly in the outline. That's one of the intracellular pattern recognition receptors, like the TLRs.
Exactly. NOD2 is a scanner inside the epithelial cells that line your intestine. Its job is to detect a piece of bacterial peptidoglycan called muranyl dipeptide.
So it's constantly seeing the gut bacteria.
It's constantly seeing them. In a healthy person, when NOD2 sees this signal from normal gut bacteria, it triggers a very controlled, low-level response. It tells the cell to produce some antimicrobial peptides to keep the bacteria from getting too close to the intestinal wall, but it also activates pathways that downregulate and suppress a big inflammatory response.
So it's a peacekeeper. It acknowledges the presence of the bacteria, but tells the system, everyone stay calm, these guys are fine.
Right. But in a significant number of patients with Crohn's disease, they have mutations in this NOD2 gene. And the mutated protein doesn't work correctly. It loses its regulatory, its braking function.
And what happens?
So when the intestinal cell sees the normal bacteria, instead of a calm, controlled response, it panics. The NF-kappa B pathway gets turned on and gets stuck in the on position.
The runner just keeps running back and forth through the nucleus.
And the nucleus just keeps screaming, fire, fire, fire. The cells pump out a massive flood of inflammatory cytokines, especially one tumor necrosis factor alpha, or TNF-alpha.
TNF-alpha. That was one of the big ones from the sepsis story.
The very same. And that TNF signal recruits huge numbers of neutrophils and macrophages into the gut wall. And those cells, doing what they're programmed to do, start releasing digestive enzymes and reactive oxygen species, trying to kill bacteria that aren't actually attacking. And in the process, they destroy the delicate lining of the intestine.
This is a perfect illustration of that core theme, regulation versus capacity.
It couldn't be more clear. The body absolutely has the capacity to dissolve its own intestine. We possess the chemical weapons to do it. But 99.9% of the time, we have the exquisite regulation to prevent it. Disease so often isn't a failure of power, it's a failure of control.
So when this control fails, how do we treat it? If the problem is too much shouting, is the solution just to gag the entire police force?
For many decades, that's exactly what we did. The cornerstone of treatment was high-dose corticosteroids like prednisone.
The sledgehammer approach.
It's the nuclear option. Steroids are powerful because they get inside the cell, go right into the nucleus, and just shut down the transcription of almost all inflammatory genes. They stop the shouting, for sure.
But at a cost.
At a huge cost, because you're not just stopping the shouting about the gut bacteria, you're stopping the shouting about everything. You're globally suppressing the immune system, so patients get side effects. Bone density loss, weight gain, thinning skin, high blood sugar, and a massive risk of real life-threatening infections.
But in the last 20 years or so, we've developed biologics, the smart bombs.
This was a complete revolution in treating Crohn's and other autoimmune diseases. Drugs like infliximab, which has the trade name Remicade, or adalimumab, which is Humira, these are what we call monoclonal antibodies.
Let's unpack that term, monoclonal.
Remember clonal expansion? How one B cell that makes a specific antibody can be grown into an army. Well, scientists figured out how to do that in a lab. They found a B cell that happened to make a perfect antibody designed to grab and neutralize that one specific cytokine, TNF-alpha.
And they cloned it.
They cloned it, grew it up in giant vats, harvested the pure antibody, and put it in an IV bag.
So you infuse these pre-made targeted missiles into the patient's bloodstream.
And they circulate through the body, acting like a molecular sponge. They hunt down and bind to every free-floating molecule of TNF-alpha they can find, neutralizing it before it can ever deliver its fire message to the other cells.
So you're not killing the immune cells. You're not shutting down the whole system. You are just jamming one specific problematic radio frequency that's causing the riot in the gut.
That is precisely it. It leaves the rest of the immune system largely intact to fight off real infections while precisely targeting the key driver of the pathology. It is true. Mechanism-based therapy.
And that is the difference between being a good technician and being a great clinician. The technician knows Crohn's equals steroids. The clinician understands Crohn's is a disease of TNF dysregulation, therefore an anti-TNF antibody is a logical intervention.
And that deep understanding, that physiological reasoning, is what allows you to predict and manage the side effects. If you're blocking TNF, what's the one infectious disease you have to be terrified of?
Tuberculosis. TB relies on TNF to form the granulomas that wall off the bacteria and keep it dormant.
Exactly. So if you give an anti-TNF drug to a patient with latent TB, the granuloma can dissolve, the TB reactivates, and it can kill them. Understanding the physiology is not an academic exercise. It directly translates into saving your patient's life.
A heavy but absolutely essential takeaway.
It is.
So let's try to wrap this incredible journey up. We have covered a huge amount of ground today.
We certainly have.
We started with this idea of the body as a crime scene. Not a fortress, but an open city requiring constant intelligent surveillance.
Then we met the innate watchmen, ancient germline encoded, using their TLR barcode scanners to recognize common microbial patterns and trigger the NF-kappa B fire alarm. They're incredibly fast, but generic. The shotgun.
Then we met the adaptive strategist. Born from a miraculous transposon accident 450 million years ago, it generates infinite diversity through gene shuffling, but it has to survive the brutal boot camp of the thymus to ensure it's safe.
We saw how these two systems integrate, with the dendritic cell acting as the crucial informant, carrying evidence to the lymph node and using the MHC hot dog bun and the 2-key safety launch to activate the T-cell generals.
And finally, through the tragedy of Crohn's disease, we saw that immunity is a conversation, a delicate balance. And when that conversation breaks down, when regulation fails, our greatest protector can become our most fearsome destroyer.
A perfect summary.
I want to leave our listener with one final provocative thought, something to chew on after they're done listening.
Go for it.
We've built this entire magnificent defensive system on 2 pillars of memory. Pillar 1 is innate immunity, which is based on ancient memory, the genetic memory of patterns our ancestors have seen for 500 million years. And Pillar 2 is adaptive immunity, which is based on personal memory, a record of the pathogens we've personally survived in our own lifetime.
Ancient memory and personal memory, yes.
So what happens when we are confronted by a threat that fits into neither category? A threat that is so novel, it doesn't carry any of the ancient barcodes the innate system is looking for. And that is so aggressive, it doesn't give the adaptive system the 7 days it needs to build a personal memory.
That is the nightmare scenario. That is the theoretical stealth pathogen, something that could walk right past the watchman because it wears no recognizable uniform and could overwhelm the city before the general even has time to clone his first soldier.
A prion disease, maybe, or something completely synthetic from a lab.
It's the conceptual gap in our armor. It's the reason the conversation between medicine and microbiology can never, ever stop.
On that rather chilling note, if you want to really see the wiring diagram for that 2-key launch sequence we talked about, please open Genome, find the chapter on immunity, infection, and inflammation. Look for the diagram showing the interaction between the APC and the helper T cell. Trace the arrows. It is 5 minutes of active reading that will solidify everything we've talked about.
Absolutely. Don't just take our word for it. Look at the map for yourself.
Thanks for diving deep with us today. Keep your cytokines regulated.
And stay curious.